This is the preparation for lecture 9. We're still working on flies. We're spending a lot of time on flies because there's a huge amount that's been known about the genetic side, so the different genes that get expressed and how they affect development, because it's a genetic model organism. And so we're going to continue along the same path. We looked at just general development, and then we looked at anterior posterior and a little bit of dorsal ventral specification. Then we looked at how some of the patterning that occurs while the egg is developing during oogenesis. And we're going to come back now to the embryo, and where we left off was that between nanos and bicoid, you had this anterior posterior gradient of hunchback protein in the anterior and caudal in the posterior, which we don't really spend much time on, but it's also there. And then we're going to look at the next step. So what happens after that hunchback gradient is generated. Okay, so this is where we left it. We've got this expression of hunchback, lots of it up in the anterior region, and very little as you move towards the posterior, and none at the posterior end. And what this does is, hunchback is the first of the gap genes. The gap genes are expressed in broad segments of the embryo, and they're called gap genes because if you make a mutation, then all of the part of the embryo that was supposed to be specified by those genes, they don't form, and so you have gaps, you have chunks that are missing in the embryo. And some of them are expressed like hunchback from anterior to posterior, or one of them is. Krupel is expressed in one band, almost dead center. And then there's NERPs and talus that are expressed in two bands, and giant here that are expressed in two bands in different locations. So we're going to take a look at these, and then these genes, these are all transient. So they're expressed only for a short time, and they're transcriptional regulators, and they basically regulate the expression of the next batch of genes. Excuse me, just about had a sneeze there. Okay, so the gap genes, they're transient, they're transcriptional regulators, and we see them in these sort of broad patterns. So here's hunchback. There's a little bit in the most posterior region, which we aren't really going to cover, but just to know that it's there. And then here's the Krupel that I was just talking about. And there are places where there's a little bit of overlap between these two genes expression. And when you combine green fluorescence with red fluorescence, you get yellow. Okay, that's not how normal colors work, but in fluorescence, that's how it works. So those are your gap genes. They're expressed in big segments, and then they regulate the expression of what are called the pair rule genes. And these are also transient, and they are also transcriptional regulators. And when we talk about pair rule, what we mean is there are 14 parasegments in the embryo that roughly translate into the 14 segments of the hatched embryo, which is the larva. And the pair rule genes are expressed in every other segment. So they're expressed in 7 stripes instead of 14. They're expressed in alternating ones. So this one is, this red one that I'm just indicating with blue, just to be confusing, is called Eve, for even skipped. And then there's odd skipped, which is, so even skipped means it skips the even one. So it's expressed in segment 1, 3, 5, 7, 9, 11, 13. And then the odd one, odd skipped, is expressed in 2, 4, 6, 8, 10, and 12, and 14. So the green is that one. So this is 2 different genes being expressed. And at this point, the embryos are not cellularized. So what you're seeing is the syncytial blastoderm. It's the embryo where the nuclei have made it, have made it to the surface. You can see the little nuclei on the surface here, but you can see that they haven't formed cells yet. So it's kind of amazing, really, when you see such a sharp boundary and you don't even have cell membranes to separate them. Okay. And then the parallel genes regulate the genes called the segment polarity genes. And the segment polarity genes, they are expressed in parts of all 14 segments. Some of them are at the anterior, some can be in the middle, some can be near the posterior, but they're expressed in this repeated pattern. And you see that there are the 14 stripes, they curl around in this way because these are being expressed at the same time that gastrulation is happening. So during the expression of the parallel genes, cellularization takes place, and then the gastrulation, the movement, remember the extension of the germ band, the germ cells are in here, they're being pushed in along with the posterior end of the gut, and then they're going to make their way to the posterior inside part of the embryo. And then here's the mouth, the little gastrulation of the digestive system, the endoderm here, and these are going to eventually form a complete tube through the body. The segment polarity genes, some of them could be transcriptional regulators, but they're expressed in a more stable way over long term. And a lot of them are signaling molecules and other types of proteins. So they're not just transcription factors the way the others are. And their expression is longer term than the others. These get expressed for a very short period of time. They turn on the genes they're supposed to turn on, and then they themselves stop being expressed, whereas these guys are longer term. Okay, so how does hunchback regulate GAP gene expression? It doesn't do it by itself necessarily, but if we look at the concentration from anterior to posterior of hunchback protein, we have these two thresholds. We've worked with thresholds and gradients before, so hopefully that will make sense. When you have hunchback that is above a certain level, it acts as a repressor of the Kruppel gene's expression. When it's below that threshold but above this other threshold, you still have some of the protein around, but it's a smaller amount. Then you get activation of the Kruppel gene's expression. And then below this amount, there's not enough hunchback to have any effect, either activation or repression. And at this point, it is in fact a different gene that sets where the posterior boundary of this of Kruppel expression is going to be. And the GAP genes themselves mutually repress each other, which we'll talk about in a minute. And so when we look at bicoid mutant embryos, so if you have embryos that come from mothers that didn't have any bicoid protein, all you have is the maternal hunchback that was put in there, but you don't get any of the upregulation. Bicoid isn't in there to induce the transcription of more hunchback. So what happens is across the whole length of the embryo, the anterior half, it's all in this region. You never have enough to be above here and act as a repressor. So you've lost this repression of Kruppel expression, and that means the Kruppel expression is expanded towards the anterior. The other gene or genes that are limiting the posterior end, those are still functioning. And so this is an example of the kind of experimental evidence that indicated what exactly hunchback is doing. When it's above a certain amount, it represses and it keeps Kruppel from being expressed right in the most anterior region, keeps it to approximately the center of the embryo. And this other gene or set of genes is keeping it sort of limited on this side. And so when we get rid of bicoid function, we also lose all the zygotic hunchback protein, and we only have that little bit that mom put in, and it's down in this amount. And so therefore, we have activation of Kruppel expression, and so there it is over almost the whole anterior half of the embryo. Okay, I had forgotten about this animation, but so at this point, when you've got below this threshold, hunchback doesn't have any effect. It's the other genes that are taking care of the posterior end of the regulation. And so if you to look at all, these are the gap genes, hunchback, which we've talked about, Krupel, which we've talked about, and these other ones, which I've just briefly mentioned. So if you were to go across the whole embryo, there would be these broad regions where, where here's NERPs, here's Krupel, here's a region in between that's just got hunchback, and here's where giant is. And then we've got tailless, a little bit of tailless here, and then a lot of tailless there. And you can guess why this is called tailless. When this gene is mutated, then we're missing the posterior end of the embryo. And if you look on the front, the first slide, this just shows you what some of the mutant phenotypes are, and you can sometimes get an idea of like why they're named what they're named. Sometimes the phenotypes, show fused segments with the little bristles are all showing, and there's none of the naked cuticle that's in between, or runt, which is very, very short, and it's missing big chunks of the segments and so on. So the phenotypes are what gives the name usually to the gene. So it's what the embryos look like when you have a mutation of the gene. And this is just a couple of pair rule genes. This is even skipped Eve, shown in the green here, and in the blue here, this is just different ways of staining in embryos to see where things are being expressed. And then Fushi Taratsu, which is definitely not pronounced correctly. It's a Japanese name that means sort of alternating stripes, or maybe it means even stripes or something like that. And that's shown in the red here, and in the brown here, 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 and so on. So this just shows you the expression patterns are really beautiful. Very satisfying to do an experiment and see these stripes come up. So the gap genes basically define where the pair rule genes are going to be expressed, and the pair rule genes are expressed in these alternating stripes. And basically, if you were to look along the embryo, there are these sections that are called para segments. They're called para segments. Para means beside, and the way the embryos are divided up with their little there's these little sort of indentations that look like sort of separators between the segments. They almost line up, but don't quite line up with the three segments in the head, the 3 segments in the thorax, and then the 8 segments in the abdomen of the late embryo. This is what the segments of the larvae will have. So the larval and adult body have these segments, and when the segments are set up in the embryo, they're just a little bit offset, so they don't line up perfectly, and we'll talk about that a little bit. There's a proposal for probably why this happens. It has to do with the nervous system, and I'll talk about that in the class. Okay. And so the pair rule genes are expressed in alternating segments, and then the segmentation genes, or the segment polarity genes, those are expressed in just part of all of the para segments. And that's shown in this picture here. So just remember, when we're talking about para segments, we're talking about the embryo, but then when the embryo is mature and about to hatch and then forms a larva, then we're talking about segments, and they don't line up perfectly. So the posterior of this segment matches the anterior of this para segment, for example. And it's just, it can be tricky to get your mind around, but once you just realize they're just slightly shifted relative to each other, then it makes it a little bit easier to understand. Okay, and then this just shows again, just to be really clear, parasegment expression. The red box is the parasegments in the embryo, and then the larva, the segments are again, as I said, they're offset a little bit, so they don't line up exactly right. Okay. After germ band retraction and the maturation of the embryo, that's when we see the actual segments. And up to that point, before that point, we see these parasegments. And they're just defined by sort of where different genes are being expressed. That's how they've been defined historically. Okay, so the parallel genes, they're expressed in these very interesting patterns. And if you think about something like hunchback, which is not evenly distributed across the whole embryo, or bicoid, or even sort of caudal, those can't be, there's no one gene, let's just say, that's being expressed in such a way that it could turn on these different stripes of expression. And it turns out that the genes themselves, the little coding regions, are very small, but they have these big, huge regulatory regions, sometimes hundreds or thousands of bases in length, because there's different enhancers. There's an enhancer for stripe 1, and then there's another enhancer somewhere else for stripe, 2 and 7 or something. I'm just making these up because the real information is coming up. But the idea is just that there are little sections of regulatory sequence, and it depends on which activators and repressors are present, whether this gene is being turned on or not. And so in any particular part of the embryo up near the anterior, it'll be probably hunchback and bicoid that are binding, and maybe NERPs or tailless or something like that are found up in the anterior part that are also part of the regulation. So it's a whole set of combinations of both the maternal genes and the gap genes that then set these really precise stripes of expression. And this just shows over time. So at the very beginning, when bicoid and hunchback are being expressed, they start activating Eve and skipped. Eve has been really extensively studied, so it's really well known. And you can see, like over time, you can see the nuclei getting smaller and smaller, so that means divisions have happened, but cellularization hasn't happened yet. And then you can see this is the timeline: earliest, later, 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 and then later, 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 and you wind up with these beautiful stripes. And probably fairly soon after these stripes get refined into these really distinct patterns, probably cellularization happens pretty soon after that. But it's kind of remarkable to see these really sharp boundaries with no actual cell membranes to divide them. And so we're just going to look a little bit at the regulation of just one stripe. This is Eve stripe 2. Stripe 2 is parasegment 3, so that's something to just keep in mind. And what we have is there is bicoid expression, which is the red line here. There's hunchback expression, which is the turquoisey line here. You've got giant up front here, and then you've got crouple approximately in the middle. And these gradations are their expression. So if you imagine bicoid and hunchback being the activators, they're going to activate Eve expression. But then if you think about giant, for example, as a repressor, it's going to prevent expression here. And if you think of crouple as a repressor, it's going to prevent expression here. So between the presence of the activating proteins and the repressing proteins, these guys repress, these guys activate. It's only in place here. The expression is going to be just in the place where you have very low, relatively low levels. This isn't zero crouple, but there's way more hunchback. So if hunchback and crouple are competing with each other, to bind to the promoter region and turn on or off, the hunchback is going to win because there's more of that protein, right? Whereas over here, giant is going to win as the repressor because it's present, it has more protein than bicoid, and so on. And so we're just going to look at this. This is just a little bit of just the regulatory region for the promoter, sorry, of stripe 2. So what you have here in this region is you have your activators, which are bicoid and hunchback, and then your repressors, which are crouple and giant. And one thing to notice is that the activating proteins and the repressing proteins are binding in approximately the same positions. It's not 100% true. You have places where there isn't competition, but in the regions where the binding sites overlap, you will have a DNA site here, and you'll have a bunch of different proteins that are vying to bind here. And so whichever ones are in the larger amount will win. If you have approximately equal amounts, the repressors will win. So if you had the same amount of bicoid and hunchback and the same, say, hunchback and crouple, just as examples, if they were about the same amount, the crouple would win, and the crouple would win because its binding affinity is stronger, so it will outcompete the activators. Okay, so there's a few things to get from this. One is that you have multiple sites, so there's lots of places along here that the activators and repressors can bind. There are certain regions where the activators and repressors are probably competing with each other, and whichever protein is in greater amount will win the battle. And then if it's more activators present, then they bind and they turn on expression of the gene. If there's more repressors present, they bind and they turn off the expression of the gene. And if you have approximately equal amounts, the repressors win because their binding is a little bit stronger. So those are the basic rules for how that works. And we'll talk about it again in the class. And then the parallel genes, like the gap genes, encode transcription factors, but and they regulate segmentation genes. And those segmentation genes are expressed in each parasegment, but not in the whole segment, just in a part of it. So these are what are called transitory groups. These aren't the real segment boundaries. They are parasegment boundaries. Those will go away after after germ band retraction happens. But you can see that in the case of this is probably engrailed. There it could be one of another ones too, but they're expressed just in the anterior part of each of the parasegments. And this basically just shows what's really interesting is this feels like a very weirdly complex way of turning on engrailed because if you look at FTZ, FTZ is the abbreviation, FTZ. And FTZ is expressed in stripes 1, 3, 5 all the way to the end. And basically the job that it does is it turns on the expression of the engrailed gene and in a row of cells, just one row of cells, and that defines the parasegment boundary. So the just anterior of engrailed is where the boundary forms. But then if you look at the next stripe, there's even skipped being expressed, and it's doing exactly the same thing. It's turning on engrailed, which is the purple, and engrailed is turned on in these cells, and they define the parasegment boundary too. So it's weird because you turn on these two different genes and then they both do the same thing, whereas you could have, you know what I mean? It seems like there's extra steps that aren't needed. But that's true for a lot of parts of development, that there are these weird things like the mother provides hunchback, and then part of the job of nanos, the only job really that nanos has, is to shut off hunchback in the posterior end of the embryo. So it's just, you know, there's these funny little extra complicated ways of doing things. That's what makes it fun. Okay. Yeah, so basically, if you go through here, these genes, even though they're being expressed in different segments, they're doing the same job. And then initially, the expression domains are only one cell wide. As the cells start to divide and the segments get a little bit wider, they will extend sometimes um, to other cells a little further back, but they begin just being one cell width. And the engrailed is really important as setting up the parasegments. And the reason is because the cells immediately ahead of it, there is this, it's not a real physical thing, but there is this inability for cells to shift or move. There is signaling that is taking place back and forth here, and we'll talk about that in the next lecture, lecture 10. But it defines what is called a compartment boundary. And this on the next segment is the other boundary. And what happens is if you do experiments and you inject and put a label on a cell inside one of these compartments, and then it divides, and then both of the cells have the label, and then it divides again, and all 4 daughter cells have the label. What you will find is the labeled cells are always between these two sort of magical boundaries. You never label one of these cells with a fluorescent dye and suddenly you see cells with the fluorescent dye in another parasegment. And that's referred to as a cell lineage boundary, because what it means is that the cell you marked and then all its descendants, which will have the same mark, and you can see the next 2 that result from cell division, they're going to have the dye, and then when those divide, those 4 cells will have it, and so on. They all stay inside these parasegments, and so that's referred to as a lineage boundary. Okay, so cell lineage restriction or lineage boundary. So a compartment is a region that contains cells. They were set aside at a certain time in development, and those cells and all their descendants form this compartment. And so the parasegment is a compartment. Okay, and that's almost half an hour, so that's enough prep. I hope though that some people have said these are making the lectures a little easier to follow and helping them with understanding. So I'm going to keep trying to do as many of these as I can.